Welcome back, students. This is video lesson 3rd in continuation with video lesson 2 based on chapter Sexual Reproduction in Flowering Plants. In this video lesson 3rd, we are going to study the structure of pollen grain and its development. Now students, we must know what is pollen grain. Pollen grain is first cell of male gametophyte. Now in video lesson 2, we studied how pollen grains are produced by the process of microsporogenesis in microsporangia and how are they arranged themselves. As we know that pollen grains are arranged themselves in the form of pollen tetrad. In video lesson 2, we studied different types of pollen tetrad. In this diagram, I have drawn two types of pollen tetrads. One is isobilateral, which is given in your NCERT book also, and tetrahedral, which is most common type of pollen tetrad. Now students, here you can see that pollen grains are arranged in the form of tetrads. Now here we are supposed to study the development and structure of pollen grain, which is first cell of male gametophyte. Now during their development, these pollen grains grow themselves and separate from each other. So you can say that pollen grains separate from each other and they grow and they increase in size and then they get separated from each other from pollen tetrad. Now here you can see one-celled pollen grain. Here you can see that it has centrally placed nucleus. Nucleus is located at the center. It is embedded in the dense cytoplasm, and here you can see that due to influx of nutrients it grows in size. Along with influx of nutrients, vacuolization resulted, or you can say vacuoles appear, which again increases the size of the pollen grain. Now because of vacuolization, nucleus shifts towards the periphery, towards the pollen cell wall. Now students, this increased pollen grain divides into two cells. Before cell division, a asymmetric spindle is formed during karyokinesis. This asymmetric spindle helps in unequal cell division of pollen grain. Here you can see that two different sized pollen grains are produced in a pollen grain. This is two-celled pollen grain. So from one-celled pollen grain, this structure becomes two-celled pollen grain. Here you can see that the smaller one is called generative cell and the larger one is called vegetative cell. Now here in this diagram I have drawn all the wall layers and we are going to study them in detail. Now here you can see that each pollen grain is covered by a sporoderm. Sporoderm divided into two. The outer one is called XII, which is made up of sporopollenin, and inner one is called intine, which is made up of cellulose and pectin. Now at certain places, sporopollenin deposition is not seen. Here you can see sporopollenin deposition is not seen. So here you can see that there is a region where sporopollenin is not deposited. It's called germ pore, from where pollen tube arises later on. Now here you can see that the larger one cell is called vegetative cell and the smaller one is called generative cell. Now this generative cell separates from the pollen cell wall by the formation of callose, and later on this callose dissolved by the help of enzyme callase and it becomes naked and it floats freely in the form in the cytoplasm of vegetative cell. It is somewhat spindle in shaped. Now here you can see that at germ pores, intine may be thickened, or you can say it has deposition of callose. Now students, here we can see that vegetative cell is irregular in shape while generative cell nucleus is very prominent. So here we have, before that let us study the function of sporopollenin. Sporopollenin is said to be one of the most resistant chemical known in this world. It can withstand high temperature, it can tolerate strong acids and alcohol, it can free from microbial action. No enzyme yet known to us that can degrade sporopollenin. That is why it is said that pollen grains can be well preserved as fossils, or more specifically microfossils. Now students, here you can see that XII may be divided into three different parts. What are the three different parts of XII? So XII may be divided into three different parts. So three different parts of XII are: inner one is continuous, inner continuous layer called foot layer. It is called foot layer. Foot layer. Then middle one is discontinuous layer called baculate. And the outermost is also discontinuous, which we call tactum. Now students, tactum provides different or characteristic sculpturing or design around the pollen grain. It may be in the form of ridges, it may be in the form of spines, it may be in the form of reticulate, etc. And these designs help expert to assign different pollen grains into different families, different genus and different species. Now here we can see that these pollen grains are now going to convert into 3-cell stage, which is said to be the mature pollen grain or mature male gametophyte. Why is it called male gametophyte? Because it is going to carry 2 male gametes. How 2 male gametes are formed? Let us study. Now students, you can see that this 2-cell stage pollen grain is going to convert it into 3-cell stage. The smaller generative cell undergo mitotic cell division by the process of mitosis. It again divided into 2 cells. Here you can see 2 cells are formed which converts 2-cell pollen grain into 3-cell pollen grain. Now students, in 60% of cases, pollen grains are liberated from anther at 2-cell stage. What are the 2-cell stage? One, the larger one, vegetative cell, and another one is generative cell. While in 40% cases, we find that pollen grains are liberated at 3-cell stage, 1 vegetative cell and 2 non-motile male gametes. These are non-motile male gametes that can be seen in 3-cell stage. And this 3-cell stage structure of pollen grain, or you can say the mature male gametophyte, is formed by 2 mitotic cell divisions. So here you can see this is the mature male gametophyte. Now the whole content of pollen grain is drop out in the form of pollen tube when the pollen grain deposited on stigma. The compatible one, say this is stigma, and the compatible one pollen grain germinate on the stigma by taking nutrients which are secreted from the stigma, and a pollen tube arises from the germ pore. From germ pores, pollen tubes arises. In monocots, you can see only one germ pore that we call germ furrows because they are elongated in shape, while in dicots, germ pores are 3 in number, and from any one of the germ pore, pollen tube arises consisting of 2 male gametes and 1 tube nucleus. So this is what about the development of male gametophyte up to 3-cell stage. Now students, in my next video, I am going to study, I am going to discuss, I am going to explain the microsporangium, its structure, its development, how megasporogenesis resulted, and how is female gametophyte development resulted. Thank you and have a nice day.